Good afternoon, everyone. Happy Valentine's Day. Welcome. I'm Jack Van Horn from the University of Virginia. It's my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. This unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since founding it in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our selected theme for this year is focused on the quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, promise, opportunities, and hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic methods, along with the wide variety of data analytic opportunities they present in data science, deep learning, AI, and in spatial modeling. We would like to remind everybody that today, Valentine's Day, February 14th, 2025, is our very last day for you to send in application materials in hoping that you might be able to join us for our Biomedical Data Science Innovation Lab, which we'll be hosting at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We're very excited for our program this year and for this 5-day mentored and facilitated activity where junior faculty, biomedical researchers, and computational experts will develop novel joint projects on topics of quantitative approaches and applications for spatial multi-omic data analytics and use those towards guiding personalized medicine. We hope that you'll submit your materials so that you might join us in Music City in June. For today's seminar, I am very delighted to welcome our speaker, Professor Hongzhe Li, who's Professor of Biostatistics, Epidemiology and Informatics at the Perelman School of Medicine at the University of Pennsylvania. Professor Li is the Vice Chair for Research Integration, the Director of the Center of Statistics and Big Data, and the former Chair of the Graduate Program in Biostatistics at Penn. His research focuses on developing statistical and computational methods for the analysis of large-scale genetic, genomic, and metagenomic data, as well as theory on high-dimensional statistics. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Professor Li's lecture today is entitled Methods for Population Scale, Single Cell, and Spatial Multi-omic Data Analysis, where he will instruct us that population level single cell gene expression and data captures the gene expressions of thousands of cells from each individual within a sizable cohort. This data type enables the construction of cell type and individual specific gene expression distributions and gene co-expression networks. And Professor Li's talk will present new statistical methods for linking such individual level gene expression distributions to covariates or phenotypes in the general framework of object data regression. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching us on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I'll be synthesizing these questions and I'll ask them on your behalf during the last 10 minutes or so of the lecture. And with that, Hongzhe, welcome. We are so excited to hear from you. We're looking forward to this presentation. Thanks again.
Thank you very much, Jack. Really pleased to be here to present this talk on method for population scale, single cell, and spatial multi-omics data. So I like to thank my collaborators, my student Hongzhe Li and Binghao Yan, who contribute to this work and NIH grant. So the motivation of my talk is on how to analyze this population scale single cell RNA-seq data. So the idea is the following. Now I think it's becoming very common that we can collect the single cell data on a sizable samples from a given population. And so for each individual, we have some covariate or disease phenotype. In addition, for some tissue or some cells, we can obtain thousands of cells for a given individual. And for each of the cells, we can have a measurement of a set of genes, 20,000 genes, for example. But different individuals, we may have different number of cells. So if you look at one single individual, if you look at one gene, we are able to study the distribution of the gene expression of that gene over cells. Or if we can look at a set of genes, we can study, for example, co-expression of a set of genes for every single individual. And the question we want to address is how to link, how to associate this individual level single cell distribution with a covariate such as age. And I think in the next several years, we're going to see many such large population level single cell data. Here is just one example, and one ongoing cohort study called the 10K10 cohort. For this study, they are planning to collect the single cell data for over 10,000 people, including healthy individuals and individuals with various diseases. In addition, they look at the blood PBMC cells, and for each individual, in average, they are planning to collect 10,000 cells. In addition, for every individual, they will also get the genotype data. So this large-scale population single-cell data will provide a lot of really important biological insight into the variability of the gene expression at the single cell level and how such variability is related to disease and other covariates. Another example is to learn the continuous cell dynamics using static snapshot of RNA sequence measurement. So here is one example. They collected the data to study the human embryonic body differentiation from embryonic stem cells. So what they did is they collected the single cells over a period of 27 days, and they measure the cell expression over 5 different time period. And one of the difficulty the goal is really try to understand the cell trajectory during this cell differentiation process. But the difficulty is there's no link. There's no link of the cells across a different time point, because once you perform the single cell measurement, you destroy the cells. So for this data set, we really want to estimate the state of the cells between this static population snapshot and to understand the cell trajectory. And this time scale are our samples. At different time, we have a sample of cells. Another example is this very recent publication by Sri Lanka in Nature, where they collect the spatially resolved single-cell transcriptomic profiling of the mouse brain process lifespan. So they look at a particular coronal section of the brain, and they collect over 4.2 million cells from 20 mouse brain at the different developmental ages, from 3.4 months to 34.5 months. And for this data set, we really try to understand the spatial cell trajectory during this aging process. So the general framework we use to answer those questions is this Freset regression framework. So just assume we have NIAID samples. So where X is some covariate, which belong to the standard p-dimensional Euclidean space. So in the previous example, X could be age, X could be time, and Y is some random object that belong to a metric space. Could be the distribution of the genes or covariance matrix of a set of genes, or distribution of the cells over different time point. So the general Freset regression framework. So here, because Y belongs to the metric space, for a metric space, we need to define a distance. Just suppose there is a distance we call it D. Then Freset mean basically is you try to find the Y, the object, that minimize the expected value of the square of the distance average over the random variable Y. So that's a standard Freset mean. And Peterson and Mueller, they further develop a Frechet regression essentially is a conditional mean. So their definition of Frechet regression is we want to define the conditional expectation y given the covariate x as a y which minimizes this weighted square distance. So here the weight S is some type of a covariate score defined by the Euclidean covariate X. And D is this metric that we're going to define. So depending on what is the Y, we're going to use a different D. But this is more general framework. And particularly if you define this covariate kernel into this very particular form, then if the Y is a standard scalar outcome, and D is a standard Euclidean distance, then this Frechet regression actually reduced to the standard linear regression model. And so besides this least square kernel, we can also use some local regression kernel, use some kernel function to define this weight. So this is how you perform regression while Y does not belong to standard Euclidean space, but in the more abstract metric space. And for the problem I mentioned before, this Y belong to the metric space. The Y could be the distribution of the gene for a given individual. For example, in the differential distribution analysis, the Y is a distribution of a single gene over samples. In the co-expression analysis, Y could be the covariance matrix of a set of genes. In the cell differential process analysis, the Y could be the distribution of the cells, where each cell is quantified by a low-dimensional representation of the gene expression. For example, for every single cell, you can represent the gene expression into 20 to 50 principal component space. Then the cell distribution basically look at the different distribution of the cells. For the spatial transcriptomic data, the Y could be the d-dimensional spatial distribution. So not only you have the PC, you actually know the location of each of the component. For all those Ys, we can use a similar framework to perform regression to associate Y with a covariate. So today I'm just going to give you a couple examples of a particular model, a particular Y in the framework of Frechet regression. So as I mentioned, when you have the random object Y, then you have to define a distance. If the Y are the distributions, then the very convenient distance to use is so-called Wasserstein distance, or the Wasserstein metric between two probability distribution, mu1 and mu2. And this distribution can be defined through this objective optimization over a T, which is called the transport map. Basically, T is a map to push the random variable from distribution mu1 to mu2. And we want to find T to minimize the cost, this L2 cost of this transport. So this is called the Wasserstein distance between two distributions. If mu1, mu2 are the univariate distributions or the multivariate normal distributions, then there's explicit expressions for this distance. However, for the general distribution of mu1, mu2, there's no closed form expression. And for the high dimensional distribution, computing this distance can be challenging. And recently, there are some new development of using neural optimal transport using neural network to approximate this optimization problem. So let me just start with the first question that we want to answer. If we want to answer this simple question, when we have population scale single cell data for a given individual, remember we have thousands of cells, even for a particular cell type and thousands of cells, this allows us to estimate the covariance matrix among a set of genes. And that covariance matrix can be used to study gene co-expression for every single individual. So you think about when we have a set of samples, we have a covariance matrix, then we can estimate from the single cell gene expression data of a set of genes. So this will be my Y. And I have the standard covariate, such as age or disease status as my covariate. So the question we want to answer, the first question we want to answer is, does this co-expression depend on covariate? Or for example, depend on age or not? And this can be important. For example, the co-expression tell us a lot about the single cell level gene regulation among a set of genes. So that's the question we want to answer. So, but now my outcome measurement is a covariance matrix, which has to be a positive definite matrix. So the space of the covariance matrix sit, they are not in the standard Euclidean space, like when you study the mean expression. So the covariance matrix of a set of genes, they actually sit into a more complex metric space with a particular Riemannian manifold structure. And that's the difficulty. So we need to have a new tool to perform regression with a covariance matrix as an outcome. And for the covariance matrix, the convenient distance to use between two covariance matrices S and Q is this so-called the Perelman distance. So you have two positive definite matrix, and you can define the distance, which has this explicit formula expressions. And this distance is exactly the Waterston distance between two mean zero multivariate normal distributions with a covariance matrix S and Q. We just use this as a way to define the distance. We do not assume any normal distribution assumption of the gene expression. And particularly for the two covariance matrix, the optimal transport map I mentioned to push from one distribution to another distribution also has this explicit expression that we can write out. And indeed, this optimal transform map minus the identity map essentially define a tangent vector of the space of positive definite matrix at a matrix S. And if you assign this tangent vector a standard Euclidean norm, then we have this Riemannian manifold structure of the Waterston space. So back to the question I want to answer. So assume now I have n individuals, and every individual have a covariance matrix Qi. For now, I'm assuming we observe this covariance matrix for each individual, and later on I'll talk about when we estimate this user data. And we have the covariate Xi. And for the Q that belong to a space of positive definite matrices, the distance we use is the Perelman distance. As I mentioned before, we're going to perform regression analysis. So my regression model, which is a conditional expectation of the covariance matrix, condition on the covariate X being little x, is defined by finding the covariance matrix that minimizes the weighted distance. And we can estimate this regression function by replacing the expectation by the empirical sample average. And so here we replace the S with a sample estimate of this covariate kernel. So my goal is I'm going to use this regression framework to develop a test to test whether the covariance matrix depend on the covariate X. So that's my goal. And before I introduce the test, I want to introduce a few notations so you can follow the test statistic I use. I already mentioned that this optimal transport map to push from covariance matrix S to Q has this explicit expression. And so this is a function of both Q and S. If you fix a Q, this optimal transport map actually is a Fréchet differentiable. In a sense, we can perform a Taylor expansion of the around the S for a fixed Q. And here the differential, this differential of this optimal transform map also has a close expression. This differential operator has a close expression that we're going to use. In addition, the Waterston distance between two positive definite covariance matrices S and Q can also be written in terms of this optimal transport map TS to Q. And this distance, this Wassler distance is also differentiable. And there's a closed form expression for the first and second Fauché differentials. And our test statistics is going to use those differential to define the test. Okay, so now before I talk about test, let me just say a little word about how to compute, how to perform this optimization, right? Because we need to find the positive definite matrix which minimize this quantity. And this can be done by a generic algorithm called the Riemannian gradient descent algorithm, originally published by Chui et al. So the idea is basically is this: you can perform the gradient descent update in the tangent space. Then you project the update to the positive definite matrix space. And so in the tangent space, remember I mentioned this T, this optimal transport map minus this identity map is a tangent vector. So this is where you perform the gradient descent. Then you map from the tangent space to the positive definite matrix space to get the final estimate. So computation-wise, this is very easy to do. Now, how do you test? So we want to test whether the covariance matrix or the co-expression matrix of a set of genes depend on the covariate X or not. So the null hypothesis would be this covariance matrix is a constant, does not depend on the covariate X. The test statistics we developed has this walled type of test statistics. So let me explain this. So in this test statistics, this is QNXI. This is a regression estimate for the ith covariate. So we have n sample, and this is covariate xi. And based on the regression, Fauché regression framework I mentioned, we can get the estimate of the covariance matrix at this covariate xi. And minus, this is a null. Under the null, we can estimate the covariance matrix. So basically, you look at the difference between each individual estimate and the estimate under the null, but you also need to apply this operator, this operator. So this operator is a Fauché differential operator of the transport map at the mean. So this is almost like a wall type of test, right? You have the difference and you have second derivative and you take a certain norm, right? So basically a way to quantify how different the individual estimate is from the estimate under the norm. So that's the motivation of our test statistics. And this H, which is a differential of the transport map, remember, I mentioned this operator has a closed form expression. So this is also very easy to calculate. Essentially, just calculate the matrix norm. That's our test statistics. So what we showed is under the null, when the covariance matrix does not depend on the covariate, then this test statistics has a null distribution of a mixture of chi-square with a degree of freedom p. So p is the number of covariate, usually small, like a few covariate we want to test. And this lambda, this weight lambda is the eigenvalue of expected value of this quantity. So this quantity can be interpreted as the covariance matrix of the error term under the null. And this is also easy to calculate because we have closed form expression for this optimal transport map. So that's basically our our test statistics. And so far, what I presented, I assume for every individual, we observe their covariance matrix. But in reality, when we analyze single cell data, we have to estimate the covariance matrix using the data that we observed. And we assume that the true X and Q satisfy the Fauché regression. But that not means when you plug in the estimated covariance matrix, the estimated covariance matrix also satisfy the Forsell regression. That's not satisfy the Forsell regression. So, but what we need is this. We only need a unbiased estimate of the covariance matrix for each individual. So each individual will use their single cell data to obtain estimate of the covariance matrix. And you assume this estimation error, this estimate is unbiased. And the estimation error satisfies some condition that are related to the sample size n. So, and they say, for example, if the data truly come from a multivariate normal distribution, then you can just use the sample covariance matrix as a QI tilde. You just plug in the sample estimate of the covariance matrix into our regression framework. That will also work. The basic idea is we can replace, for every individual, we can replace their true covariance matrix QI with the estimated covariance matrix, and the test still perform well. And it really due to the unbiasedness of our estimate, the number of cells, which is M, is allowed to be smaller than the sample size. When the sample size is large enough, the number of cells has to be bigger than square root of the sample size, but can be smaller than the sample size. So this is basically our test statistics. I'll just show you one plot. So we did a lot of simulation evaluations by generating some data. So here is one set of simulation result. I just want to point out a few things. So I'm assuming, for example, I have total of 200 individuals. So this is N is 200. Assuming I have three covariate, so I have three X covariate, I want to test whether there's association with a co-expression. And then now I consider D, like for example, a small set of 10 genes, and I use our test, apply our test, and run the simulation 200 times. What you see here are so this is obviously it's a low dimension, it's a 10-dimension case. And assuming for each individual, the number of cells is small. Like if you only have 50 cells, this is the first plot, or 100 cells, or infinity means you actually observe the covariance matrix. What you see here is the empirical distribution of the test statistics against the theoretical distribution, the theoretical null distribution, which is a mixture of chi-square distribution. So when the number of cells is small, the test is under the null, the test statistics pretty much still follows the theoretical null distribution, a little bit deviate here for a few runs. When we increase the number of cells to 100, you see now the null distribution agree with the empirical null distribution. We agree with the theoretical null distribution pretty well. The third plot is the setting where we assume the covariance matrix is known. In other words, you assume the number of cells is very large. So I think there's another issue when we plug in the estimated covariance matrix into our regression framework. However, what we do observe is the reduction of the test power. Remember here, we want to test whether there's association between a covariate, this three-dimensional covariate, with the covariance matrix of 10 genes. And what you see here, the solid. So here so first look at the this blue line. So this blue line, the top one is assuming we observe the covariance matrix, and the dashed line means we have to estimate covariance matrix using the single cell level data. And you can see if the number of cells is small, the power is indeed reduced quite a bit. That's because even for 10 genes, if you only have like 50 cells or 100 cells, the estimate of covariance matrix could have large variability, and that translates into smaller power of the test statistics. However, we compare our test with another test statistics called the distance-based covariance test. So basically, for every two individuals, you calculate the covariance matrix, then you can compute the distance. What is the distance? Then based on the pairwise distance, you can perform a test called the distance-based covariance test. You can see in that case, the D covariance co-test always has a smaller, lower power than our proposed test, right? Even when the covariance matrix are observed, if we estimate the covariance matrix, the power from this distance-based covariance test is much lower than our proposed test. So this is just one example I want to show you. Basically, emphasize that if the number of cells is small, then estimating the covariance matrix can be very noisy, and that actually translates to lower power of testing association between the covariate and the covariance matrix. So in order to derive the null distribution of this test statistics I talk about, we need to obtain the uniform convergence of this regression estimate. And we obtained this in our paper. In addition, in our paper, we also obtained the central limit theorem for the regression estimate of the covariance matrix for any given covariate X. And this is very useful because if you give me a covariate X, I can estimate the covariance matrix. And because of the central limit theorem, I can perform a test for every pair of genes I and J. I can ask whether their covariance matrix or co-expression depend on the covariate X or not. And so this is useful for single cell level co-expression analysis. So let me present a few examples of how to use this test. So the data we look at is a data set published in this science paper in 2022. Basically, in this study, they collect the PBMC single cell RNA-seq data from a total of 982 healthy individuals with age range from 20 to 90 years age. And we look at a particular set of genes, 61 genes that belong to this so-called nutrient sensing pathways. And this pathway is related to insulin action. And the pathway has been shown to be related to aging and have been shown to be somewhat dysregulated during the aging process. For this PBMC cells, first we identify a particular cell type called the naive central memory CD4 T cells, and we only look at the cells of this particular cell type. For the whole study, they have a total of roughly 1.2 million cells. And when we limit ourselves only to this particular cell type, then for some of the samples, the number of cells of this particular cell type is small. So we further reduce the number of individuals from 982 to 734 individuals with at least 200 cells. Then we look at those individuals. Every individual, some of the genes, this is 21 genes. Some of the genes they have zero variants. Basically, they are not expressed in some of the individuals. Obviously, we cannot study the co-expression of those genes. So this further reduce the 61 genes that belong to this pathway to 37 genes. So 37 genes, every individual, we have at least 200 cells. So what we did is every individual, we estimate the covariance matrix for each of the individual using the UMI library size scaled count, and with a batch correction, use the standard software SRAD. So we normalize by correct the library size. Then we obtain the expression for the 37 genes. at each of the cells for each of the individual, then each individual compute the covariance matrix. So the question we want to ask is, does the gene co-expression of the 37 genes change with age? How does co-expression change when people get older? So that's basically the biological question we want to answer. So what you see here is a plot of the age. So x-axis are the age, and y-axis are the eigenvalue of the covariance matrices. First three eigenvalues. So this dashed line are the eigenvalue obtained from the raw covariance matrix, right? So every individual, you can estimate covariance matrix, then you compute the eigenvalue. So those are the based on the observed estimated covariance matrix, the eigenvalues. For the top three eigenvalues. The line is not quite a linear line, but this is very close to a linear line. Is the for every given age, remember, use our regression model, we can obtain the estimated covariance matrix. Then use the estimated covariance matrix, we calculate the eigenvalues. And you can clearly see this the first two eigenvalues, you see they go down as people getting older. And this line, this is the eigenvalue based on our estimated covariance matrices at each of the time point. Looks like a linear line. Actually, there's a interpretation why this look at the linear. Is essentially, if you look at the estimated covariance matrix, if you look at the eigenvectors or the PCs, they are actually pretty close. They are similar. So in a sense, most of the changes are at the eigenvalue level. And if you apply our test, we got the p-value of 0.0012, indicating the association between the co-expression matrix and the age. And as I mentioned, for every given age, like 20, 30, 40, all the way to 90, we can obtain the regression estimate of the covariance matrix. And based on the central limit theorem, we can also perform a test for every pair of the genes to test whether the covariance between two genes change as a function of time as people are getting older. So at any given age, for example, at the age of 20, and here what you see here, the plot, the heat map actually are the p-values. The darker line means more significant covariance when we test whether the covariance is being zero or not. Darker line means statistical significance, lighter lines no significance. And you can see, 20, 30, 40, 50 years, they're roughly similar. But once you reach age of 60, you see a much fewer connection among this set of genes. So much sparser co-expression among the genes after age of 60. There are obviously some variability from the data, but in general, seems like age 60 is an important age where we see a larger dysregulation among this set of 39 genes. And we look at this, we think about why this is the case. I think one interpretation is if you just look at this particular cell type, like CD4 positive T cells, the idea is during the aging, this cell get repeatedly stimulated by different antigens. And this lead to highly differentiated T cells with newly acquired functional capabilities. And this can lead to aberrant expression of the genes. And that, I think, can explain why we see a less regulation among a set of genes when people getting older. So this is one example. As I said, so far I just present this population scale single cell data really allow us to estimate individual specific gene co-expressions, and use this for set regression framework. We can test we can perform regression with a co-expression matrix as an outcome. And there are many other possible applications of this idea of optimal transport map and the first set regression for population level single cell data analysis. I'll briefly talk about another application, which is a first set regression for the cellular distribution over a particular biological process. So here's the problem of studying the cell dynamic based on the snapshot of RNA-seq data. So the data we have is we have a time point during a given biological process. We have n time points and denoted by TI. And at any given time point, we have a collection of cells, a large collection of cells. And for every cell, for a given time point I, for a given cell J, we can represent this cell by a low dimension representation of the gene expression. Usually you just do a PC, principal component reductions. Then D usually range from 20 to 50 dimension. That usually is a good dimension to capture the variability of the gene expression. So at a given time Ti, what we have is the distribution of the cells, right? Over all the cells we observe at that given time. And so that's the observation we have. Now, when we perform regression, now my X are the age, are the time, a different time, right? So I can similarly perform for set regression, but now instead of using this regression kernel, we're going to use the standard regression kernel weight as my weight. And here, the distance we use is what is the distance or so-called unbalanced optimal transport map-based Washington distance. So the regression basically, for any given time point, use the observed time point, I want to estimate the distribution of the cells, the cellular distribution in this PC space. So my regression estimate are the regression. So this is the most difficult, the difficulty with this first set regression is the computation, right? Because you want to find a d-dimensional distribution which minimize this weighted distance. And that's actually computation-wise is very challenging. I'm not going to show you too much detail, but I'm just saying this distance, you can use a standard optimal transport map based on Washington distance, or use this unbalanced optimal transport distance. Now I'll skip this unbalanced OT distance, but just briefly talk about the algorithm we developed is based on this generative neural network. Essentially, we estimate a different push-forward map from a latent variable to the center, to the regression that we want to estimate. And we're going to use neural network to model that push-forward map. And for any given sample, observed sample cell distribution, we estimate another map to map from the regression to the observation. So after we have this optimal transport map, then we can use the map to follow how the cell goes as time goes. So that's basically our idea of performing for set regression of the distribution of the cell over time. So as again, I said, the computation is the most difficult part of this project. The formulation is very similar to before. Essentially, you want to find a distribution which minimizes the weighted Washington distance. So let me show you a couple applications of this regression model. So this is the data set I mentioned before. Is basically you can look at the single cell expression during the embryoid body differentiation. Remember, they have 5 time points. And what we did is we can leave one time point out. We can leave each of the 3 time point out and use our regression model to generate the cell, to estimate the distribution of the cell, use the other data point that we observed. Okay. So what you see here, for example, if you look at the evaluation for day 7.5, what you see here is the 2-dimensional PC plot of the cells that we observed. So those are the truths. The red color are the cells that we observed. The blue color are the cells that are generated from that distribution that we estimated for 4 different, methods. So this is based on unbalanced OT. This is OT distance. This is the first 2 column are our method. Another one called MIOLO, another one just based simple average, like a baseline average. It's hard. So the idea is if the generated blue dot overlap with the red dot, that means you generate the cell well, right? The cell you generated from your model is similar to what you observed. It's hard to read here, but you can summarize how well the generated cell will agree with the distribution of the cell that you observed at each of the time points based on different quantity. This is basically 3 different measurement of how close the generated distribution with the observed empirical distribution. And in general, you can see our regression-based method gave a smaller error, smaller error. So that seemed to work. Essentially, we use the data observed in multiple time points to perform regression to estimate the distribution of the cells. So, and as I mentioned, after we fit the regression, we can estimate the optimal transport map. And that actually really allows us to follow as time goes, how cells going to move from the initial time point to the last time point. You can see there's a cluster of cells that start at a similar place. They follow this trajectory, and at the end of the observation, those cells essentially follow this trajectory and move to this location. But there's another class of cells. They tend to move, not directly go to this cell at the last time point. They tend to move in the space to other state. Then, so they have some intermediate state. Then they move to the last time state. So basically, the optimal transport map really allow us to follow each of the cells and see how they move during this particular developmental process. So this is, I think, is interesting and is useful. Another data set we look at is the cell feed, also cell differentiation of progenitor cells into the blood cell lineage analysis. So here they collect the single cell expression data over 4 different days during the cell difference progenitor cell differentiation, and so we use our regression. First thing is we can estimate any cell state for any time point, not just the observed day 2, 4, and 6, because we performing regression. And also we can track how the cells goes during this time period. You can see a class of the cells, they basically follow this path and it ended up to here. And another set of cells, they can move to another type of cells. So the regression can capture that in the regression framework. And this is similarly, we did this check, leave the cell data from day 4 out and use our regression to estimate what the cell looks like for day 4. And you can look at the choose cells and the model predicted cells, same like our method, again, give the best fit of the cell distribution. So now we just use this regression to study this time sequence of single cell measurement. And this regression model really allow us to estimate to generate the cells for any time point between the time that you have observation. So that's one use of this regression model. Another one is the trajectory, because we have this optimal transport map. We can push the cell from one state to another to understand the trajectory. So anyway, I think my time is almost up. I think the population scale single cell data really allow us to study the individual variability of distribution of the genes across the different cells, even the cells of the same type, we observe a large variability. And this allow us to study the association between individual level covariate or outcome with the single cell level variability of one single genes or the cells or a set of genes if you study the co-expression. And this optimal transport map and the first set regression provide a framework to study this type of association, because the outcome now is not a standard Euclidean observation anymore. It's belong to a certain metric space. And this neurogenerative model, which I didn't show you the details, help us to solve the computational difficulty. One project which I don't have time to present is we are also trying to extend this idea to the population level spatial omic data. Jack, I think this is all I have. Thank you so much for listening.
Hongzhe, that was fantastic. What a tour de force treatment of this, very unique sets of data that you've been working with. I want to thank you for really bringing a level of mathematical rigor, but also understanding of an appreciation for the underlying biology. So often with, you hear from our statistical colleagues and our data science students, and they are just applying methods out of a box, and they don't really seem to understand what the underlying biology is. So I want to thank you for, making sure that at every step of the math you're doing, there is a purpose that, can be mapped back to the biology to help give you that explanatory power. Because I feel that sometimes we miss that. So thank you.
Thank you.
One of the things that you talked about is, sometimes, like, for example, you talked about the article from Sun and colleagues where they had like 4.2 million cells, but they only had like a sample size of 20 animals. What do you suppose with some of these methodologies, is there a curse of dimensionality here that we have to respect? What recommendations do you have for people who are gathering that type of data and want to apply these types of methods?
Yeah, Jack, that's a really great question. So, having a lot of cells that really enable you to study the cell-to-cell variability and the cell distribution. For example, you mentioned Sun's paper at any given age, developmental age, because you have so many cells, then you can truly understand for example, gene co-expression. You do need to have a large number of cells. So in that sense, for a given individual or given time point, more cells better to study the distribution, because the whole idea of my talk is really to look carefully for a given sample, the variability or distribution of the gene across the cells of the same type. But now, people are getting more samples. So here's the 20. They try to cover, most of the developmental ages. Our method, you can still fit a regression model with 20 observations. And we are not at the point of making any inference yet with a 20 observation. But I think you can still fit the model, even for the time course data, because we are fitting the local regression. Most of the weight you put in there are the samples that are close to the time that you want to look at. So, and the other way to deal with dimensionality, for some of the problem, we reduce the dimension. First do the PC if you only care about the cellular distribution. I think that will work. For study co-expression, that's a major limitation. You correctly pointed out. We cannot study co-expression of thousands of cells at the individual level because for a given individual, we don't have that large number of cells. So somehow we have to limit ourselves to a smaller set of genes. And that's why for our analysis, often we look at a set of genes defined by some prior biology. Another way is you somehow filter the genes. But that's a limitation. Yeah, for a given sample, if the cells is small, it's hard to study a large set of genes in terms
Yeah,
of
I imagine it's a little easier when you're dealing with experimental animals and they, basically
PBMC cells.
yeah, their genomes are pretty homogeneous, right? And you're making one or several manipulations. It makes it a little easier. When you're dealing with a study like that paper that was it Nature or Science by Yazar and colleagues?
Science.
That one seems like you've got 982 individuals, 61 genes, that you boiled down to 37 genes, but you got 200 cells. I took notes. And that, obviously making population level inference in, just like humans or a wild type kind of organism, that's going to be a little more challenging.
I agree. Zach. That's it.
The one question I had I liked the thing that you showed at the end there, where you were tracking the eigenvalues over time,
Mhm.
and you were showing those three curves as they tracked over time. And as I wrote down my note about it, that was, do the eigenvectors of those still kind of align with each other? Because obviously the eigenvalues tell you one thing, but the eigenvectors tell you the directionality. And you seem to allude to it that they do sort of all track in the same direction. So it's really the eigenvalues which are changing. Did I understand that correctly?
Yeah, yes. Because I was really curious about why this, because our model is not linear at all. Remember, we directly model the covariance matrix.
Yeah.
Turn out this eigenvalue almost linear. You see here, and if it's linear, then I think we actually did some plot of the eigenvectors, and they are, if you look at the angles eigenvector, they are quite similar across a different age. So I think the main difference is driven by those eigenvalues.
Yeah, that's interesting. I'd be curious if the eigenvalues, if those, time point to time point, that angle doesn't differ too much, but if they're on some sort of a trajectory that's actually, rotating over time. So the individual, time point to time point, the angles are small, but it's actually because it's actually going through some trajectory. You'd be the perfect person to figure that out.
Yeah, that's a good question. We should take a closer look at that. It might be interesting.
And you mentioned, by the way, in one of your earlier comments about the noise in affecting the statistical power, and I'm curious if if you have the facility to do it, whether or not doing a principal components analysis at one stage of your modeling, if you can't cut out, like for example, with in a factor analytic approach, where you're only keeping the large eigenvalues that seem to be accounting for variance, and you get rid of the rest. If there's a way to do some kind of feature selection in your PC space like that, does that help?
Well, first thing is that you cannot reduce the data to PC because we really want to look at the individual gene level co-expression, right?
Right.
But I think what you are saying is if you make some further assumption on the covariance matrix with a small number of cells, you make a more precise estimate of co-expression, and that can lead to, because this is what you say here, this is a sample covariance matrix.
Okay.
So we didn't make any assumption to the structure of the covariance matrix. For example, if you impose a certain structure, could be, if assume all the covariance matrix, they share the common eigenvectors, then you can get a very good estimate because now you can use all the cells across all the individuals to estimate the common eigenvectors. Then you just estimate eigenvalue. So if we feel comfortable to make that assumption, so that's one approach. Another approach, I think you are saying, for example, if you assume the true covariance matrix are somehow sparse, which most likely, and with that assumption, you may get a better estimate. Then with a better estimate of the covariance matrix, you would expect a better power of detecting the association. Yeah.
Well, Hongzhe, this has been really intriguing and thought-provoking work. It's like so mathematically elegant as well. So thank you so much for sharing this. And I think it's really great that you've demonstrated the true power of being able to blend mathematics and statistics and data science with a biological challenge like these single cell expression patterns. I think this is fantastic work. Thank you so much for sharing it with us today. Everyone, today is the last day to submit your materials for our Biomedical Data Science Innovation Lab. I hope that any of you junior faculty working in spaces like this, for example, would contribute your materials so that we can take a look at you, and hopefully we'll see you in Nashville. In the meantime, everyone have a wonderful Valentine's Day with the people that you love, and we will see you next time. Thank you again, Hongzhe. We really
Thank you
appreciate
very
it.
much.